I will be describing a new optical sequencing method which can be used for rapid biomedical diagnostic by taking single molecules of your DNA, RNA, and even proteins and trying to sequence all of these biomolecules at a single molecule level without making any copies. First of all, why do we need such a method? This is primarily to combat this imprecise nature of our medicine. Nominally, these are the 10 highest grossing drugs in the US, and for every person that they help here in blue, there are a lot more, anywhere from 1 to 3 to 1 to 24 people that it is unable to help. So all these 24 remaining people either show no effect or negative side effect because the drugs are prescribed primarily on the basis of the symptoms rather than any true biomarkers that could help us diagnose diseases, health, and human wellness. In order to come up with this, first we need to understand then why do these drugs sell so much. So I thought I would quote a philosopher who best described it by saying, the art of medicine consists in amusing the patient while the nature cures the disease. So why would we develop a new sequencing method? First, the current sequencing methods rely on taking a copy of your DNA and then making millions to billions copies of that DNA. Now, while copying, there could be lots of errors. You could lose vital information like nominal changes in the DNA letters A, T, G, and C. And all of this can only be done for DNA and increases the complexity, cost of the method, making it inaccessible to everyone. So what we are trying to do is just take a copy of your DNA and simply sequence it by reading it from one end to the other. The second question that you may ask is, then why use light? Because with light, you can massively parallelize things without developing specialized electronics or anything expensive, and all you really need is very simple metallic substrates. So the real reason is to come up with low cost, rapid biomarker diagnostic tool. And we are trying to now squeeze light into tiny single molecules into nanometer scale mode volumes, which are roughly 1/1000th of the thickness of human hair or 1/4000th of the thickness of human hair, and volumes that are a billion times smaller than the smallest microscopic particle that you typically may have seen under a microscope. So we could do this by using light scattering, and nominally light falls on something and comes out with the same color. It's simply scattered, but a very small fraction of that light actually ends up giving its quantum energy to the molecular vibration. If we can cut down the large fraction of light that falls on a material and simply look at this very small fraction, we can come up or directly measure the molecular vibrations, which are the true chemical identity of the molecule. And we can really do this by surface enhanced spectroscopy using these tailored or well-designed tips, which we can now make billions of tips on a single wafer or a substrate or tiny little nanoparticles. Now, when you take these nanoparticles and put these molecular vibrations pretty close to it, these nanoparticles or tips act like an antenna and amplify these signals several fold, so you're looking at a much stronger signal. Now, if you take two such positively charged nanoparticles and add a tiny fragment of your DNA in between, it makes kind of a nano sandwich. And when we take a laser light and irradiate this tiny little nano sandwiches, for example, in solution here, you can eliminate the light that has the same wavelength or color and only look at light that is characteristic of the molecular vibration. And the spectra looks something like this, where we want to get the genetic information from the spectra about this tiny little fragment of the DNA. Now, how can we do this? And here's an actual live recording of this nano sandwich DNA and looking at the Raman spectra, and you can see it's fairly bright, and you can see like this red light coming out, or green and yellow light coming out. So the more green light may be corresponding to an adenine, and the amount of red light may be indicative of the thymine content. So here we are squeezing light not to single letters, but a small fragment of the DNA, which may be 5 or 10 DNA letters. So we've developed these optical fingerprints for different DNA nucleobases like adenine, guanine, cytosine, and thymine, referred to as AGCT, by using first the backbone of the DNA, which is common, and using it as an internal calibration standard, and then using characteristic molecular vibrational peaks to use it as an optical sequencing method. When we calibrate these signatures, you would see that as the content of this adenine, or the green light in this given block, decreases, you will see an equivalent decrease in the block content, or as the content increases, you will see more and more Raman scattered light coming in from adenine molecular vibration, which gives us a characteristic calibration curves for all 4 DNA nucleotides. Now it becomes fairly simple. We could take tiny fragments of DNA. Here I'm showing fragments taken from a pathogen which is antimicrobial resistant, and here we have taken only 12 little fragments from the entire genome, and these fragments are coming in from an antibiotic resistant gene sequence. And when we take these fragments, let's say a tiny little fragment shown here, just made up of 10 DNA nucleotides, you can take this measurement and come up with the adenine, guanine, cytosine, and thymine content very quickly, leading to a rapid identification of the block content. So we have about 20% adenine, or 2 adenine in this block of 10, and only 1 thymine based on this rapid measurement, which could be taken in a matter of few seconds, as you saw in that video. Now if we take the content of these 12 DNA fragments, here I'm showing 15 in here, this is fairly representative of very rapid measurements, and you will see that we do get some errors here and there. If I get error in guanine content of any fragment, it'll amplify and lead to a 20% decrease in the accuracy, but overall we got a good high accuracy of 93% from this very rapid measurement in real time. And as seen here, as a comparison between actual and predicted content, we saw that primarily the errors were coming in from the guanine content. So why would we use now block contents instead of single letter sequences of biomolecules? And this is a very simple problem, much like trying to stream movies or content over the internet using YouTube or Netflix, rather than taking gigabytes full of CD drives or Blu-ray discs. Now, all the content, even in a high def television, can easily be viewed by first packing that content using a lossy or a lossless data compression method and filling up the space, and then unpacking that data to quickly recreate the entire movie in high definition. Much in the same way, this block optical content collapses the data of 10 DNA nucleotides into a tiny content, and then we unpack it by comparing this data to a gene database. Here's this rapid identification algorithm, or unpacking of the data that we measure very quickly using the optical sequencing. So once we have this content, we take the block content and now we unpack it by comparison to a gene database. So if we are looking, for example, at pathogens, we would look at all possible multi-drug resistant pathogens, and there's a whole category of 4,000 multi-drug resistant genes. And when we compare it, we can assign scores based on a simple read of this block, and you will see that as we read more and more blocks, we can quickly rule out possibilities that have fairly low scores, and then come up with rapid identification of multi-drug resistance and pathogens involved. Let us show you this with the 12 tiny blocks that we read. So using this low coverage, we can unpack the data. So we read all of these contents, and if we start comparing with each block read, block 1 all the way to block 12, on how this compares to all these drug resistant genes, and you have about 4 to 5,000 curves over here, you can see that the correct curve or the correct gene here is identified very quickly in simply 5 reads. And not only the multi-drug resistant pathogen, which could help us in determining the right antibiotic regimen to be prescribed, but also the pathogen, and we could identify Pseudomonas here very quickly in about 8 reads. So with these 12 tiny reads, which could be made in a matter of seconds, what we've shown is with much less than even a single coverage of the entire genome, with less than 15% coverage of the gene, we could quickly identify anything of relevance for biomedical regimen identification. We can prescribe the correct antibiotic and correctly identify the pathogen here. And you require anywhere from 10 to 100-fold coverage, thousands of times higher coverage in typical commercial sequencing methods. So I hope I've convinced you here that by using these nano sandwiches and ramen blocks, or either in these pyramidal tips, we can rapidly identify the gene sequence here, and we have much more than 15 fragments over here, and we can not only predict the block content but then unpack this information by comparison to a gene database and quickly arrive at a picture that's relevant to biomedical diagnostic by either identifying the drug resistant genes so the correct antibiotic could be prescribed, or a personally tailored treatment plan which forms the basis of personalized medicine. So I'd like to conclude by thanking Lee, Sagar, and Amia, which are the students that did all of this work, and the funding agencies that really made this work possible.